I'm Denise McKibben, and we are at the Western Connecticut Health Network Biomedical Research Institute. Our team is focused on validating novel diagnostic procedures for detecting cancer by looking for rare cancer cells circulating in blood, for Lyme disease, which we are looking for the bacteria that causes Lyme disease in blood. And these are our main two focuses right now. Danbury Hospital is a very up-and-coming and progressive place to work. It's a friendly atmosphere, patient care is at the top priority, and up-to-date treatments are the focus of the majority of our research.
We would see lines coming from the top called B lines.
The community has been very generous in donating finances for us to perform this research, in participating in our studies, and we are really trying to give the community answers to all of the questions surrounding Lyme disease and cancer. So I started out as a chemist. I am from New Milford, Connecticut, which is a neighboring town to Danbury. I was inspired by a high school chemistry teacher and loved chemistry. I went to undergraduate school for chemistry. But when I finished undergraduate school, I really wanted to be more in tune with helping people directly. So I took an immunology course when I was working at Johns Hopkins in a biology lab and got interested in immunology. And then I went to graduate school to study immunology, in which I studied diabetes and other immune-related diseases. I worked for a pharmaceutical company, Regeneron, for a little while, and I took 8 years off to be at home with my kids. And then I really wanted to come back and do research. As luck would have it, Dr. Fiedler was starting his pathology research group at about the same time that I started wanting to come back to a career. And he was very excited to have somebody come and start tinkering in the lab and working on his assay. So the slides are inserted into here. Start with that. The current testing measures for Lyme disease are not very good at detecting it when you first start to feel ill and you might want to go to the doctor. So we have a collaboration with a company called RareSight, and they're based in Seattle, Washington. The instrument that we have that is built by RareSight, it's called an AccuSight scanner, is not widely available yet. Danbury Hospital was one of the first research institutes to obtain one of these instruments. The beauty of the machine is to concentrate a large volume of blood and all of the cells of interest into a very small volume, so then that small volume can be analyzed. The hope is that by the time you are feeling ill and start to have a fever and aches and pains, that we would be able to take a blood sample and use that blood sample to possibly detect the bacteria and say yes or no, do you have Lyme disease at that point? Because the earlier you get treatment for Lyme disease, the higher the success rate of completely eliminating the disease. This could lead to more effective cures for Lyme disease, more effective therapies, and actually perhaps new vaccines to treat patients before they would get the disease. The main cancer that we were focused on designing a detection method for is multiple myeloma, and more so a disease that's a precursor to multiple myeloma known as MGUS, multiple gammopathy of undetermined significance. For MGUS, there really are no symptoms. The patients don't feel sick, but a high percentage of these patients that have MGUS go on to acquire multiple myeloma, which is a cancer. So most patients will have to undergo a bone marrow biopsy, which is quite a painful procedure. We are trying to develop a blood test such that we could avoid bone marrow biopsies for these patients. The assay itself is fairly quick. It's a matter of drawing blood from the patient and a couple hours in the lab to measure the number of cells. Okay, so this is a slide that was analyzed for tumor cells for our MGUS project. And this is an example of all of the tumor cells that it found on that particular slide. This is the nucleus, the DNA, and then 3 different proteins on the surface of the cell that we're looking for each of the cells that it found. I perform scientific research to look for answers and to, in the end, help people alleviate suffering. We are driven to work hard to develop a new technique such that these important diseases could be detected earlier, more efficiently, and lead to better care and treatment for these diseases.